Did you know that during dialysis, osmosis is also taking place? In a regular dialysis, there is a decrease in plasma osmolarity. These changes in plasma osmolarity determines the shifting of fluid between intracellular and extracellular compartments.
But what is osmosis? Osmosis is the movement of solvent from a low to high solute concentration. One good example is dialysis.
Dialysis is when kidneys no longer function anymore. Dialysis keeps the body in balance by removing waste, salt, and extra water to prevent them from building up in the body.
There are two types of dialysis, hemodialysis and peritoneal dialysis.
In hemodialysis, blood is removed from the body and filtered through an artificial semi-permeable membrane that allows small objects such as water, salts, and metabolites to pass through, yet not allowing larger objects such as proteins and blood cells.
Isn't it so interesting? On the other hand, peritoneal dialysis utilizes the actual lining of the abdominal cavity. It is used as the filter and the solution is ejected directly into the abdomen. As blood flows around the peritoneal cavity, diffusion of solutes from the blood occurs directly through the peritoneal membrane, and then the fluid is removed later in the form of urine.
Dialysis machines use the power of osmosis to get toxins out of the blood and replace the filtering function of the kidney.
Do you want to know some examples?
Sure, let's find out!
Suppose 15 milliliters of 20% glucose solution was put in a dialysis bag. After 25 minutes of immersing the glucose bag in a beaker of water, the volume of the bag became 25 milliliters. Given a temperature of 37 degrees Celsius, we were asked to calculate for the osmotic pressure.
First, we calculate for M2 using the formula M1V1 is equal to M2V2. We assume 100 grams solution where 20 grams is glucose and 80 grams is water. Given the molar mass of both glucose and water, we convert grams to moles. Since molarity is mole solute over liter solution, we convert the given volume from millimeters to liters. Substituting the given values, we get 0.015 moles.
Finally, we solve for the osmotic pressure. Osmotic pressure is equal to the Van 't Hoff factor of 1 times molarity, the constant R, and temperature. Substituting the given values, we arrive at the osmotic pressure of 0.3689994,725 atm or 0.37 atmosphere.
Now, for another example, we have a 9.0 milliliters of sample protein and 0.510 moles sodium chloride was dialyzed against 1,000 milliliters of buffer. After the system attained equilibrium, the 9.0 milliliter sample was transferred into a vessel containing 1,500 milliliters of fresh buffer. What was the concentration of sodium chloride after the second dialysis was performed?
In order to obtain the concentration of NaCl in the second dialysis, the first one should be calculated. Given that the sample protein is 9 ml and the concentration in molarity is 0.510 m, we could calculate the concentration in molarity after the first dialysis.
First, we should convert 9 milliliters into liters, which would give us 0.009 liters. It should be the V1 or the volume of the concentrated solution. On the other hand, the M1 or concentration in molarity of the concentrated solution would be 0.510 moles in our formula. After it was dialyzed, the V2 would be 1.009 liters when we add 1.0 liters of buffer and 0.009 liters.
Substituting the values would give us 4.549058474 times 10 raised to negative 3 m or 4.55 times 10 raised to negative 3 m as the concentration in molarity after dialysis or M2.
Now, We would compute for the concentration of sodium chloride in the second dialysis. We would utilize 4.54 or 4.55 times 10 raised to negative 3 moles and 0.009 liters as the M1 and V1 respectively. After the second dialysis, the V2 would be 1.509 liters. When we add 1.5 liters and 0.009 liters, substituting the values would give us 2.713, 156,148, or 2.71 times 10 raised to negative 5 moles as the M2 or the concentration of sodium chloride after the second dialysis was performed.